cell is going to divide. So in a dividing cell, the nucleus will have chromosomes, and in a interface nucleus you will see chromatin. This is the main difference, because this is going to pack up nicely in the form of a small structure called as chromosome. Now a chromosome generally carries chromosome always carry, when you talk about a chromosome which has undergone growth, so, or you can say the S phase. During S phase, the amount of the DNA is going to double. That's why you are going to see two chromatids together. Otherwise, a normal chromosome is supposed to carry only one chromatid. After S phase, in next chapter it will be clear to you, it is going to double its amount of DNA. So now you see two copies of the same structure. Now these are two chromatids which are joined at one primary constriction called a centromere, and each chromatid has two arms. There are secondary constrictions also possible. For example, the nucleolus organizing region is controlled by 13th, 14th, 15th and 20th and 21st chromosome. These chromosomes carry a secondary constriction called as NOR, and these constrictions are responsible for the location and position of the information of the nucleolus inside a nucleus. Chromosomes can be of four types on the basis of the position of the primary constriction. If the primary constriction is at the center exactly, making both the arms equal in size, it is called metacentric. If the primary constriction shifts slightly further, or you can say closer to the one end, you'll find one arm is smaller, the other arm is longer, you will call it as the submetacentric. If it is further moving towards the corner, now the one arm is extremely small, the other one is longer, it is called acrocentric. And when the primary constriction is at the end, so only one arm is there, it is called as telocentric type of chromosome. Then we talked about the cytoskeleton. I told you the various types of cytoskeleton elements. Cytoskeleton is a supportive framework, and it is made up of microtubules which are hollow but non-contractile, unbranched, and made up of tubulin, alpha and beta type. And I told you regarding the microfilaments, which are the smallest element in a, or smallest component inside a cell. But remember, the microfilaments are not found in the plant cells. And they are solid, they are unbranched, and they are made up of actin filaments. Actin plays an important role in the muscles. So in that chapter of the skeletal system and locomotion of human physiology, we will learn detail about this concept. And intermediate filaments are those which are in between the size, and they are hollow, they are non-contractile. Remember, microfilaments are contractile in nature. So please remember, all these components of the cytoskeleton, they are supportive they form a supportive framework, and these all are proteinaceous in nature, and they form a network inside the cell so that they can regulate the distribution and orientation of different organelles. And these filaments are responsible, or these elements are responsible for all intracellular transport, cyclosis, which is called the streaming of the cytoplasm, basically the cytosol, which is always the endoplasm, and provides the mechanical support and maintains the shape of a cell. Remember one thing, that this topic we are going to understand in more detail, because we are going to learn now about cilia and flagella as well as the centriole. So just remember that the size of the microtubule is 25 nanometer in diameter. It is hollow, so the cavity inside is 15 nanometer. So the leftover size is 10 nanometer. So on both sides, 5 nanometer is the thickness of the wall. And please remember that the microtubules exist in the form of 13 longitudinal dimers, or you can say 13 longitudinal dimers of the tubulin. So when you see a long section, a long view like this way, what you see right now in the diagram is like this, a circular thing. When you see it in the longitudinal manner, you will find that the microtubules are made up of tubulin which are running in a spiral manner, and made up of alpha beta repetition of dimers. These dimers are called as the protofilaments.